1. Hi, my name is Joseph, and I'm a third year majoring in computational and systems biology with a minor in data science engineering. I've been an undergraduate researcher for Dr. Pei Ping for around 2 years, but for the past 2 quarters, my main focus has been automating the collection of experimentally identified proteins within biomedical text. As for a deeper introduction, this lab has a large focus on implementing machine learning models and deep learning-based information extraction systems, as well as using advanced data structures such as knowledge graphs to uncover hidden relationships between proteins and diseases in the vast amount of scientific literature available online. One of the first steps of this entire process is collecting the proteins to search against the publications. This lab focuses a lot on hard proteins, specifically extracellular matrix proteins, which, in order to find what these proteins are, we usually start with some query in a mass spectrometry database, such as the Proteomics Identifications Archive, or PRIDE archive. Then we gather the relevant publications associated with these mass spectrometry experiments, since the results of these experiments are usually listed somewhere in the text. However, this step is currently usually only done manually. People essentially have to look for whatever supplementary file contains some identifier for the proteins they want to gather. And this is where my arm of research comes in, since we want to automate this process to reduce efforts in a time-intensive and laborious task. Later on, we will have all the proteins from the relevant experiments we want. We can then input these into some text mining pipeline and then analyze the results accordingly. I hypothesize that by using some named entity recognition, or NER techniques, it is possible to automate the collection of proteins reported in biomedical publications with high accuracy and efficiency. As for the materials and workflow, in automating these pipelines, these were my following steps in the workflow. The first step was to collect these proteins we want to search against the text. In this case, we just wanted to search up heart proteins in general among humans, rats, mice, and pigs. So our only criteria was for a mass spectrometry experiment to have heart as a keyword in the PRIDE archive. Continuing with data collection, once we find the relevant PRIDE identifiers, we can then find the corresponding PubMed identifier, or PMID, to gather the relevant text programmatically. Following this data collection step, we can then use various named entity recognition methods. BioBERT is a commonly used transformer model that is typically used with natural language processing, or NLP, tasks that require some models based on large biological text-based corpuses. spaCy is an open-source Python library with various NLP capabilities. Its submodule SciSpaCy was used for the purposes of this project and was trained on the BioNLP13CG corpus. Finally, PubTator, a tool developed by NCBI to automate the annotation of biomedical texts, was also used. Once these entities were collected, we attempted to map them back to UniProt IDs. Prefix trees were used to search against a curated list of synonyms, and cosine similarity was also used to determine if the collected entities could be mapped back to some UniProt identifier. As for the results, in terms of actually seeing which of the three models work best, PubTator and spaCy seem to work relatively well in conjunction compared to BioBERT by itself. However, it is important to note that the BioBERT model and tokenizer use the 1.1 version created by the Data Mining and Information Systems Laboratory at Korea University. It is very much possible that a primary reason for why BioBERT seems to be underperforming against the two other models was because only pre-trained weights were used. In examining many of the entities that BioBERT collected using pre-trained weights, most entities were only one word long and sometimes had the first few letters truncated. PubTator and spaCy, with default settings, were mostly able to gather phrases as well as single words. Upon closer inspection of the entities collected by both, PubTator was usually more successful in the collection of abbreviations for entities, whereas spaCy was able to typically get the full spelling of those abbreviations. Mapping these entities back to some UniProt identifier was also a non-trivial task in this research project. A prefix tree was used in an attempt to match collected entities against a manually curated synonyms list. Cosine similarity was also used to determine if a given entity had a high similarity to any words in the curated synonyms list. After completing the mapping, it was shown that cosine similarity was able to match these synonyms better compared to prefix trees. 70.6% of these entities collected from the aforementioned methods actually ended up finding at least one match when using cosine similarity. This is a significantly better finding than the 22.6% that prefix trees were able to match. In addition, prefix trees were not able to get any perfect matches. However, cosine similarity, while still at a relatively low percentage, was at least able to get 6.45% of a perfect mapping, which essentially means that it found an exact match between the two words. While cosine similarity tends to produce more results, it is also worth noting that it is significantly slower than using a prefix tree. For the future, looking into how BioBERT should have its pre-trained weights is one possible avenue of exploration. There are also other NER paradigms aside from BERT, such as conditional random fields, which may provide substantive results. In addition, there are various other ways of computing string similarity between entities and synonyms, such as Levenshtein distance, or finding the overlap coefficient. In conclusion, when looking at the results between PubTater, spaCy, and BioBERT using default settings, PubTater and spaCy were usually able to collect a significant amount of entities with a higher accuracy compared to BioBERT. In addition, when mapping the entities collected, cosine similarities seem to provide better mapping capabilities compared to prefix trees. While this project still has a long way to go in terms of examining the full capabilities of these NER models, I hope you were able to see the value in researching methods of automating protein curation. Thank you.